type 1 and type 2 diabetes are a major cause of morbidity and mortality in today's society. As a result, many companies are trying to generate pancreatic cells, and specifically beta cells, from a variety of stem cells and a variety of stem cell sources. This is patent that was recently issued, which discusses ways of making stem cells that are non-pancreatic to differentiate into the pancreatic differentiation pathway and hence possibly be therapeutically useful. What we're going to be talking about is not legal or medical advice, and we make no warranties as to the validity of the statements. Essentially, the background of this patent is that stem cells are being considered as a treatment for diabetes. A wide variety of different stem cells have been used in animal models, and also some have entered clinical trials for treatment of diabetes. Hematopoietic stem cells are in clinical use for a variety of different indications, and of course, bone marrow transplantation is an example of hematopoietic cell transplantation. Mesenchymal stem cells are very far along the clinical development pathway, and they are in phase 3 for indications such as graft versus host and Crohn's disease. The patent essentially covers ways of making pancreatic tissue or pancreatic leg tissue from hematopoietic and mesenchymal stem cells. If we look to the claims of the patent, the first claim begins by covering a method of transdifferentiating mammalian non-pancreatic stem cells selected from hematopoietic and mesenchymal stem cells to enter the pancreatic differentiation pathway. Now, if we continue reading the claim, it comprises of culturing the said stem cells in a media that permits the expression of pancreatic differentiation markers. So, the media it's stating that the media permits expression. It's not saying that the media actually induces expression. So, maybe an interesting point. But anyway, the media which are covering in the claim is essentially it's a DMEM media with high glucose and sodium pyruvate using human or bovine sourced albumin, serum albumin, tumor captoethanol, which acts as an antioxidant, fetal, calf, or human serum, as well as antibiotics, insulin, transferrin, and selenium. Now the claim continues and it gets broken down into hematopoietic stem cells versus mesenchymal stem cells. It states wherein the said hematopoietic stem cells are cultured for at least 14 days in the medium, plus they add certain factors, stem cell factor, GLP, activin, and so on. All of these factors mentioned have been implicated in one sense or another to be associated with stem cells and also with pancreatic differentiation. For example, EGF and gastrin have been used in clinical trials for inducing pancreatic differentiation in vivo. So they're covering these factors, but also the conditioned media of a cell line. And they are claiming these three cell lines, CAP and ARP and AR42J. Basically, conditioned media means that you culture the cells, the cell lines mentioned, collect the media which contains a variety of different things, and then add that in combination with the growth factors. Now, if we continue reading, Claim 1 continues to cover the mesenchymal stem cells for differentiation. Mesenchymal stem cells are cultured for 21 days in said media, plus now they repeat some of the growth factors mentioned for the hematopoietic stem cells, and also conditioned media. Claim 2 covers the method of Claim 1, wherein the pancreatic markers that are expressed are selected from the group consisting of PDX1, ISL1, PAX4, 6, GLUT2, CK, cytokeratin 19, and insulin. So basically, if you look how they're covering this, they're not saying that they're making islets. Well, and they're not saying that they're making cell useful for therapy. What they're saying is they're making something that expresses some of the markers and these markers are associated with pancreas. So basically they're covering what they're making in claim 2. In claim 3, the inventors are covering what they are making it from. So they say the method of claim 1, wherein hematopoietic stem cells are CD34 positive, and in claim 4, they define mesenchymal stem cells as CD105, 166, 29, and 44 positive. Claim 5 says that they're making the stem cells from the human. And claim 6 is an interesting claim. It's the method of claim 1, but a method of producing an endocrine hormone. So not a method of producing a cell or differentiating cell, but a method of producing a hormone, comprising the method of claim 1, and further comprising the step of continuing to culture said transdifferentiated cells in said media, whereby an endocrine hormone may be produced. So I'm curious how these claims would be interpreted, but they seem to be pretty interesting claims, claim 6. Now if we look at the data, figure 1 of the patent essentially shows culture of hematopoietic stem cells in the basal media plus table 1b growth factors. We're going to show you these at the end. And what they're showing is day 14 of culture of these hematopoietic stem cells was able to induce expression of several transcription factors and genes associated with pancreatic differentiation, such as PAX4 and 6 and cytokeratin. And if you look at day 45 of culture, you can actually see some insulin being expressed and cytokeratin being expressed. And on the second figure of the patent, they're using a mixed culture of hematopoietic stem cells and mesenchymal stem cells for 14 days in the basal media alone. And again, they're showing induction of a variety of different transcripts associated with pancreatic differentiation. Table 1a, these are the components of the basal media that they used to generate the previous data. And table 1b, these are the factors that they added, factors that they claimed. So it's very interesting because there's another figure also, the figure showing a picture of the cells, but it didn't come out very well. So this pattern is very interesting because essentially it's teaching how to produce some cells which appear to produce insulin and have some characteristics of similarity with cells of the pancreas. Thank you very much.